Good morning, all of you. Myself, Janvi Sahu, student of B.Ed first semester from St. Thomas College. So, today we will study about the topic sexual reproduction in flowering plants. So, in this video lecture, we will study about the introduction, structure of a flower, and then we will study about the stages of sexual reproduction, in which we will cover pre-fertilization, double fertilization, and post-fertilization. So, let's see what is a flower. Flower, a fascinating organ of angiosperms. Flowering plants are also known as angiosperms and they use a sexual mode of reproduction. Reproduction in plants mainly revolve around the flower, which consists of both male and female gametes. All parts of a flower aid in the process of reproduction. So, to understand the process of sexual reproduction in flowering plants, we should first understand the structure of a flower, including its various parts and their functions. So let's see structure of a flower. First, the calyx. Calyx is the outermost hole of a flower. It comprises units called sepals. In the figure provided, we can see the green colored part. So in the bud stage, calyx encloses the rest of the flower. Usually exhibit green coloration. At some other instances, they may be colored. Calyx can be prominent or absent. So as we have seen in many flowers, there are calyx present, but in many they are absent. Now the second one is the corolla. It's the second hole of the flower. It consists number of sepals. So in the figure provided, there is a hibiscus flower which consists of the corolla or the sepals in the red color. So petals are sometimes fragrant. They are colored, thin and soft. They help in the process of pollination as it attracts animals and insects. So now the reproductive structure of flower. In this we will cover androecium and gynoecium. So first androecium. It is the male reproductive part of a flower and it's the third hole. It comprises of stamen and each stamen consists of two parts, which is anther and filament. They can be seen in the diagram. So the anther is the bilobe structure, whereas the filament, it is a long slender stalk-like structure. The tip of anther is supported by the filament. Here, pollens are produced by meiosis and they disappear eventually. Now the second is gynoecium. It is the female reproductive part of the flower and it's the fourth and the last hole. It comprises of pistil, which includes stigma, style and ovary, and this can be seen in the diagram. So stigma, it is the landing platform for the pollen grains. Then style, it is the elongated cylinder part beneath the stigma. And the ovary, it is the basal bulged part of the pistil. The ovary produces ovules internally. Now, on the basis of pistil, gynoecium can be of four types: monocarpellary, multicarpellary, syncarpous and apocarpous. So monocarpellary means the gynoecium has a single pistil. For example, peas and beans. Then multicarpellary means gynoecium which consists of multiple pistils. For example, strawberry. Then syncarpous, it means gynoecium that has combined pistils. For example, tomato and cucumber. And the last one, apocarpous, which means gynoecium having free pistils. That is, for example, lotus. So now let's study about the stages of reproduction. So the stages of sexual reproduction includes mainly three stages, that is pre-fertilization, double fertilization and post-fertilization. The pre-fertilization includes various structures and events including microsporogenesis and megasporogenesis. And post-fertilization deals with events in endosperm and embryo development and conversion of ovules into seed and ovary into fruit. So let's study pre-fertilization. First, the structure of anther. Anther is the breeding part of stamen and it is typically a typical angiosperm anther is bilobe with each lobe having two theca, which means they are dithecous. Each anther is connected by two lobes, each lobe comprising of two pollen chambers located longitudinally. It is responsible for pollen formation. Anther is kept in place by a filament which is thread-like structure attached to the anther as we have seen before. The transverse section of an anther shows tetragonal structure consisting of four microsporangia located at the corner 2 in each lobe. So here in the figure we can see the transverse section of an anther, and microsporangium develops further and become pollen sac. Now the structure of microsporangium. In a transverse section, a typical microsporangium appears near circular in outline. It is generally surrounded by 4 layers that are epidermis, endothelium, middle layers and tapetum. These layers can be seen labeled in the given diagram. So the first 3 layers, that is epidermis, endothelium and the middle layer, they perform the function of protection and help in dehiscence of anther to release the pollen. And the last layer, that is the tapetum, its main function is that to nourishment and development of pollen grains, and it is the innermost layer. When the anther is young, a group of compactly arranged homogeneous cells, which are known as sporogenous tissue, occupies the center of each microsporangium. Now, what is microsporogenesis? As the anther develops, the cell of the sporogenous tissue undergo meiotic division to form microspore tetrad. Each cell of sporogenous tissue is capable of giving rise to a microspore tetrad. Hence, each one is a potential pollen or microspore mother cell. Hence, this process of formation of microspore from a pollen mother cell, that is PMC, through meiosis is called microsporogenesis. The microspore, as they are formed, are arranged in a cluster of 4 cells, that is microspore tetrad. As the anther mature and dehydrate, the microspore dissociate from each other and develop into pollen grains. Inside each microsporangium, several thousand of microspores or pollen grains are formed that are released with the dehiscence of anther. So as in the figure we can see the dehiscence of anther, there are a number of pollen grains. Now the structure of pollen grain. Pollen grains represent the male gametophyte. They are generally spherical, measuring about 25 to 50 mm in diameter. Now, a typical pollen grain consists of 2 layered walls. First, the exine, and second is the intine. Exine is the hard outer layer of the pollen grain. It is made up of sporopollenin. It has prominent apertures called germ pores where the sporopollenin is absent. So here sporopollenin is the keyword. It is the most resistant organic material till known, which means it can withstand high temperature and acid alkali. No enzyme is as far known that can degrade sporopollenin. Now the second layer of pollen grain, that is intine. It is the inner layer of the pollen grain. It is thin and continuous layer made up of cellulose and pectin. The cytoplasm of pollen grain is surrounded by a plasma membrane. When a pollen grain is mature, it consists of 2 cells, that is vegetative cell and generative cell. So vegetative cell, it is bigger in comparison to generative cell and has abundant food reserve and a large irregular shaped nucleus. Whereas the generative cell is smaller and it floats in the cytoplasm of the vegetative cell. It is spindle shaped with dense cytoplasm and a nucleus. Then structure of megasporangium. So the megasporangium, that is ovule, is a small structure attached to the placenta by means of a stalk called funicle. The body of the ovule fuses with funicle in the region called hilum. These terms can be seen in the figure, labeled in the figure. So the hilum represents the junction between ovule and funicle, and each ovule has 1 or 2 protective envelopes called integuments. Integuments encircle the nucellus except at the tip where a small opening called the micropyle is organized. Now opposite the micropylar end is the chaliza, representing the basal part of the ovule. Enclosed within the integuments is a mass of cells called the nucellus. Cells of nucellus have abundant food reserve, and located in the nucellus is the embryo sac or female gametophyte. An ovule generally have a single embryo sac formed from a megaspore. Now megasporogenesis. So the process of formation of megaspores from the megaspore mother cell is called megasporogenesis. So ovules generally differentiate a single megaspore mother cell, that is MMC, in the micropylar region of the nucellus. It is a large cell containing dense cytoplasm and a prominent nucleus. the MMC undergoes mitotic division. So in the diagram, we can see the first one ovule showing a large megaspore mother cell. Then the second one a dyad of ovule, and the third one a tetrad of megaspore. So now comes a question that what if the MMC undergoes meiosis? As in this slide we studied that the MMC undergoes mitotic division. So what if the MMC undergoes meiosis? Meiosis will result in the production of 4 megaspores, but as we know only one megaspore becomes functional later. So now the structure of embryo sac. Embryo sac represent the female gametophyte. In the majority of flowering plants, one of the megaspore is functional while the other three degenerate. Only the functional megaspore develops into the female gametophyte, that is the embryo sac. The method of formation of embryo sac from a single megaspore is termed as monosporic development. There is a characteristic distribution of the cells within the embryo sac. So, three cells are grouped together at the micropylar end, which is seen in the figure in the below part. It is the micropylar end and it constitutes the egg apparatus. The egg apparatus in turn consists of two synergids and one egg cell. The synergids have a special cellular thickening at the micropylar tip called filiform apparatus, which play an important role in guiding the pollen tubes into the synergid. Now, three cells are at the chalazal end and are called antipodals. They can be seen in the figure in the above part. The central cell has two polar nuclei, and a typical embryo sac at maturity is 8 nucleate and 7 celled, which means they consist of 8 nucleus and 7 cells. Now, what is pollination? So many of us have heard about this term before. Pollination. It is the mechanism to achieve objective of fertilization. So the transfer of pollen grains from the anther to the stigma of a pistil is known as pollination. Various agents of pollination include abiotic agent or biotic agent. So pollination can be done either by biotic agents or abiotic agents. Abiotic agents include wind and water, whereas biotic agents includes animals and insects. So majority of plants use biotic agents for pollination like bees, butterflies, wasps, ants, birds. They are common. Then wind pollination occurs if the pollen are lighter in weight, and water pollination is quite rare. Then comes fertilization. It is basically the post-pollination event. After pollination, when the right type of pollen, which means compatible pollen of the same species as the stigma, arrives, the pistil accepts the pollen and promotes post-pollination event that lead to fertilization. If the pollen is of wrong type, which means if the pollen is non-compatible or the pollen is of different species of the stigma, then the pistil rejects it by preventing pollen germination. The pollen grain germinates on the stigma to produce a pollen tube through one of the germ pores. The contents of the pollen grain into the pollen tube. The pollen tube grows through the tissue of the stigma and style and releases the ovary. Pollen tube, after reaching the ovary, enters the ovule through the micropyle and then enters one of the synergids through the filiform apparatus. And all these events from pollen deposition on the stigma until pollen tubes enter the ovule are together referred as pollen pistil interaction. Then comes double fertilization. It is the second stage. So after entering one of the synergids, the pollen tube releases the two male gametes into the cytoplasm. One of the male gametes moves towards the egg cell and fuses with its nucleus, thus completing the syngamy. This results in the formation of zygote. The other male gametes move towards the two polar nuclei located in the central cell and fuses with them to produce primary endosperm nucleus, that is PEN. This is termed as triple fusion. Since two types of fusions, syngamy and triple fusion, take place in an embryo sac, this phenomenon is known as double fertilization. Now, this event of double fertilization is unique to flowering plants. The central cell after triple fusion becomes the primary endosperm cell, that is PEC, and develops into the endosperm while the zygote develops into an embryo. Now, post fertilization. It is the last stage of sexual reproduction. So, following double fertilization, events of endosperm and embryo development, maturation of ovules in seeds, and ovary into fruit are included in post fertilization. So, first, endosperm. Endosperm development precedes embryo development. The primary endosperm cell divides repeatedly and forms a endosperm tissue. The cells of this tissue are filled with reserved food materials and are used for the nutrition of the developing embryo. In the most common type of endosperm development, the PEN undergoes successive nuclear divisions to give rise to free nuclei. Then subsequently cell wall formation occurs and the endosperm becomes cellular. The number of free nuclei formed before cellularization varies greatly. For example, coconut. The coconut water is nothing but free nuclear endosperm and the white kernel is the cellular endosperm. Now, endosperm may either be completely consumed by the developing embryo before seed maturation, for example, pea, groundnut, beans, or it may persist in the mature seed, for example, castor and coconut. Then embryo. Embryo develops in the micropylar end of the embryo sac where the zygote is situated. Most zygotes divide only after a certain amount of endosperm is formed. Stages of embryo development, that is known as embryogeny, are similar in both monocotyledons and dicotyledons. Now there are two types of embryos on the basis of number of cotyledons. So the first one is the dicotyledon and the second one is the monocotyledon. So, a typical dicotyledonous embryo consists of an embryonal axis and two cotyledons. The portion of the embryonal axis above the level of cotyledons is the epicotyle, which terminates with the plumule or stem tip. The cylindrical portion below the level of cotyledons is hypocotyle that terminates at its lower end in the radicle or root tip. Root tip is covered with a root cap. A typical dicot embryo is given in the figure. Now the monocotyledons. Monocotyledonous embryo possess only one cotyledon as the name suggests. In the grass family, the cotyledon is called scutellum that is situated towards one side of the embryonal axis, that is the lateral side of the embryonal axis. At the lower end, the embryonal axis has the radicle and root cap enclosed in an undifferentiated sheath called coryza. The portion of the embryonal axis above the level of attachment of scutellum is the epicotyle, which has a shoot apex and a few leaf primordia enclosed in a hollow foliar structure, the coleoptile. This is also provided in the figure, a typical monocot embryo. Now, the seed. The seed is the final product of sexual reproduction. It is often described as a fertilized ovule and they are formed inside the fruit. A seed typically consists of seed coats, that is the cotyledon, and an embryo axis. So, mature seeds can be of two types, non-albuminous or albuminous. So, non-albuminous have no residual endosperm, for example, pea or groundnut. And albuminous, it retain a part of endosperm, for example, wheat, maize, barley, etc. So, occasionally in some seeds such as black pepper, remnants of nucellus are also present and this residual is the perisperm. Integuments of ovules harden as tough protective seed coats. The micropyle remains as a small pore in the seed coat. This facilitates entry of oxygen and water into the seed during germination. And as the seed matures, its water content is reduced and seeds become relatively dry. The general activity of the embryo slows down and the embryo may enter a state of inactivity called dormancy. As ovules mature into seeds, the ovary develops into a fruit. That means the transformation of ovules into seed and ovary into fruit proceeds simultaneously. The ovary wall develops into the fruit wall called pericarp. The fruit may be fleshy, like we have seen guava, orange, and mango, and it can be also dried, like groundnut and mustard. So that's it for this video. Thank you.